عليكم ورحمة الله وبركاته. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. من بعد إن شاء الله تعالى we'll be continuing with week 4 for practical C17, practical 40 of BIOC17, Microbiology here at UTSC. And inshallah ta'ala, students will do their plaque assay, streak plating, transformation, conjugation of bacteria. So stay in tune, inshallah ta'ala, and let's do.
Yes.
Yeah. Or if I haven't marked you in for attendance, just let me know. I try to keep track of it for everyone. So in case you find that it's not the case, even in general, just let me know. So, okay. Yes. You will get them short, or in due time. So let's start off with a few points. So I finished your unknown assignment for marking purposes. I have to wait, as you probably saw from my announcement yesterday, I have to wait from Dr. Brunt to get the approval for you guys to see it on Quirkus. However, if you want to have a look at your grades or receive your flowcharts slash data sheets with all the percentages and values, like your score, then let me know and you could take them in between or at the end of today's practical. A few other things. I may or may not be able to come next week, and that would mean then I would probably see you after reading week. So just bear that in mind. I'm not feeling too well. So if, of course, you want to stay away, that's perfectly okay. If you want to ask questions as well, you can ask them at your benches, and I'd be more than happy to help accordingly. Uh, Hopefully everyone also saw the announcement, right? Has anyone not seen the announcement? Please raise your hand. And it's okay if you haven't. I could even show it on the, let's see if it connects. It may not actually connect here, but essentially, let's see if I can go on with this. It's just very important that you guys, like, I know it's sometimes hard to tell, but I send them individualized. Let's see. I send them as like essentially these updates, the ones that you see here. You send 30 individual emails? Like individually? Well, they send out individually to you all. So like yeah, I actually want to clarify that. So I do take time to write this down at nighttime. I know it's sometimes it's a bit late, but often because I'm doing too many practicals myself that it's a bit difficult to navigate. Okay, who am I teaching? So nonetheless, these are some key tips I want all of you to think about. And very importantly, as you can see, number one, punctuality. I know for today it's excusable because of the winter conditions outside, but just bear in mind for future notes that you want to come in as soon as you can. Also very important, notebook quality reflects your lab performance for pretty much what you put in is what you take out or expect to leave out with throughout our practicals together. Meaning like if you show me very few details and limited effort in your lab notebook, then that may actually reflect your performance because you may not know actually what you need to do. for each exercise. So try to bear that in mind. I also note that some of you included timings, which is really good. So bear that in mind. When I was in undergrad, I also would do the same. So it's a good tip to keep you well prepped for step 3 and keeping track in time. So we have actually 2 hours and 15 minutes to complete the lab. So what I'll do is go straight away uh, to the PowerPoint. So let's go. Any questions on me? Questions? Okay, we'll just continue as it is now. Streak plating, which will be done in the first 15 minutes, is pretty much a similar technique of what you've done for spread plating, except you're now dealing with. The idea is that you are essentially streaking bacteria along a certain quadrant or quadrat, and you are essentially repeating that process, diluting your bacterium from one location to another. So you just want to obviously label, right, your plate with your name. This is individually done, by the way. So your name, you can just write it down, or your initials, the practical, which is 8, the date as well, and the exercise, because we're dealing with multiple plates. So please make sure that you refer to the exercise. This one's exercise 1. The date, I believe, is February 6th. February 6th. And then essentially, then yeah, your initials, and it could be on that side as well. Now, the same applies for your other plates. However, you're going to include your bench. So just keep that in mind. Group 1, 2, 3, 4, 5, 6. So when you're working in groups of 4 or 3, you'll just bear those points in mind for your upcoming plates. Any questions? No questions? No questions? So to simplify this question, a pure culture is pretty much a culture, be it on a plate, on an agar slant, that essentially represents one bacterial species. So let's say it's Micrococcus luteus, Micrococcus roseus. If they are found together, that's actually a mixed culture. So that's something you want to keep in mind. And in comparing spread plating, which we did last week, which is like you doing pottery or baking a cake and moving your agar plate with your bacterium all around, right? Evenly distributed. In this case, you have them as shown here for streak plating, essentially a streak or passed through the agar with your colonies at different locations throughout, distributed differentially throughout your agar plate. The benefit of this is that you're actually diluting your bacterium, which you can see through the colonies becoming more visible down to the very end here. So that's something that you want to keep in mind, because for future experiments and especially transformation for today, you'll be taking out 1 to 2 colonies for preparing your conjugation experiment. Sorry, your transformation experiment. Any questions? Any questions? Okay, so streak plating, we're gonna do this for the next 12-ish minutes is okay as well. Very simple. I just mentioned it right now. But you want to first label your plate with the 4 points I mentioned earlier, and then the quadrat itself, which looks like a T, that green T in the middle, right? And then that way you'll be able to separate your quadrants and start off, by the way, with one edge likely towards the edge of a plate, but it could be just essentially at your first quadrant and then eventually connect it back to the other quadrants or quadrants. Now, yeah, that's what I would say. I pretty much answered, if I'm not mistaken, this as well. You can also think about how you would answer this question. It's really with regards to isolating your bacterium, very important as well for future use, and what we'll be doing shortly, transformation. So let's go straight ahead. If there are questions, let me know. I don't mind to slow down. But let's go straight ahead. I'm gonna make this 12. This should actually take 5 minutes, but I'm just like, no pressure. 12 minutes. If you finish early, the better it is. The gas is on as well, so if you want to begin with your group, or sorry, individually, you can do so. We go to the upcoming exercises. I'll talk a bit more as to what you need to do next. But you should have Micrococcus luteus either on your benches or in the middle bench here. If you want to share Micrococcus luteus, you can do it by groups of 4. So you can take it if one member of the group hasn't taken it so already. You don't have it. I'll let you know. I'll wrap it. In the meantime, the plates that you have on your benches are for the other exercise. Please keep note the NYC plates for the track acting, the conjugation one, sorry, the manuscripts or the chemical, these plates, dark or golden brown, maroon and golden brown. Then I'll get the plate for it. In the meantime, you can try I just debated like what are you doing? On the plate, so what we're going to do is go straight to conjugation. That will take more time, and that's okay. So because we're going to be incubating in about 2 hours, we might as well make this focusing on that. And then following incubation, we can take care of exercise 1, 3, and 4. Yeah, absolutely. I was there. These are plates for exercise 1. So they just came in. So it's up to you if your group wants to focus on exercise 2. I would recommend, I'll just say that at the podium. I would recommend going with exercise 2 for now because it's more time sensitive. We have to actually place it into the incubator for 2 hours, meaning that we want to take care of this exercise in the next, I would say, 20 minutes. So don't worry about the timer right now. I'll adjust it accordingly, but focus on exercise 2, and then it should be okay. I'll go straight ahead to exercise 2 just to give you some insight as to what you need to do. Sorry. I meant exercise 4. So I was saying exercise 2 referring to conjugation, but Exercise 4 is conjugation. So sorry for the confusion. Let's just say it one more time. So I'm going to ask you if everyone can pay attention. It's one of the final slides of today's practical, but it's very important for you to be able to understand what you need to do here. You'll be working as groups for this practical, so just bear that in mind, groups of 3 to 4, right, within your block of each bench. So remember what you're starting off with. You have 3 plates, one golden brown chloramphenicol, rapamycin is also golden brown, and then the chloramphenicol plus rapamycin are essentially maroon. So you have 3 plates. You want to, once again, label the plates with the 4 traits I mentioned earlier: practical, exercise, your group, and the date. And then make sure that you make a line essentially between both sides, because you should have both bacteria, HT99 and J53R, on your benches. They're on white trays, so have a look. You should have white trays with tubes labeling HT99 and J53R. The one with a black lid is the one that you're going to use for conjugation of the 2 bacteria. Or bacteria. So bear that in mind. When you reach this step, okay, let me ask, how many of you are doing conjugation exercise 4? All of you? Okay, I just want to make sure, because I don't want anyone just going into other exercises without direction. So please bear in mind the following. You've labeled your plates. You've made, you cut them in half, or like you labeled half sides with HT99 and J53R, and that you're essentially following the steps shown here. You're first off placing with the sterile swabs. I'm just going to show it with one of the benches here. You should all have. You have perfect. Just read it up. Read it up. Show it to everyone. Those are the swabs you're using. Don't use the other ones that are on your in the white boxes that are in Ziploc bags. The ones that have hashes indicate that they mean autoclaved and they're sterile. So just bear that in mind. When you're taking them out, try to take them out where the hash is located, so that way you can take them out and just gently place. They should be liquid, if I'm not mistaken, the J53R and HT99. So essentially, you add in with the swab into each of the tubes separately, separate swabs, right? And then aseptic technique once again, because you're flaming the neck of each tube accordingly. When you do this, or after you do this, once again, bear in mind your bench is sterile, 70% ethanol. You're going to repeat this process, you're going to add in that swab into one location. It could be HT99, depending on your bacteria, or J53R. And then that's where you're just following simple smearing, what you've done in the past. Last week was spread plating, except you don't need the L rod. You just are literally smearing or doodling around your plate with that single swab. And that's what you're doing for the 1-drop spread. You're spreading it around from that single swab drop. And that's done for HT99 and then separately for J53R. So don't worry about the other steps, because you need to do that for the first 2 plates. And you can have someone in your group just have essentially pipette 0.1 mils with the sterile pipette tips on your benches, the green boxes, 0.1 mils of HT99 and 0.9 mils of J53R. This shouldn't take too much time, but the sooner you do it, the better. So I would say these 2 steps can be done in 10 minutes. The last one, to make it easier, another 10 or even 5 minutes to do. And if anyone's struggling, just let me know. But now you can begin turning on your Bunsen burners, label your plates,
like as group members, take on different roles, and you can also begin taking or adjusting your pipettes. Use the P1000 for 0.9 mils and P200 for 0.1 mils, or you can use the same pipette of a P1000 for both volumes. If you have questions, let me know. I could slow down accordingly. Yes, label them with the agar side on top, so how it appears as inverted. You can label your plates on that side. Group files. So another thing some of you were asking, or some of you may be wondering about using pipettes. Make sure as well, Chana, that you flame the neck of your tube. Yeah, perfect. So, do you mind if I show? When you're done with the black lidded conjugation mixture of HT99 and J53R, you can label it, and we'll be adding them into the incubator together. Let me clarify. You have different instruments for different exercises, everyone. The ones that are like the small epi tubes, the tubes that are pipette tips, this is for your class assay. So bear that in mind. Your ice block for transformation, and what you have on the tube rack for
So we're heading inside the 10 to the negative.
yeah. Okay. Don't worry about the label itself, but you will label it. Okay. Let's just. So, okay, let me just say something very important, because it's obviously different from last time because of the situation with exercise one. I didn't mention something very important. Please make sure you resuspend your bacteria, right? Because your tubes are going to have a pellet at the bottom for HT99, J53R, and any other tubes, glass tubes that you're using today. So bear that in mind. Make sure you resuspended your bacterium. such that you flick them multiple times, because you're going to need to do it not only for now, but following incubation for 2 hours. If you don't do it right, you're going to have a poor yield for your plates. So please make sure that you do this, bearing in mind what's also shown in the manual. But just to reiterate this, you flick both of HT99 and G53R before adding them in. You resuspend them. When you've labeled them, you can place them. We'll place them into the incubator. But don't forget, you have also the other 2 plates to do these swabs. To do sterile swabs I mentioned earlier with your Bunsen burner on, so that way you reduce contamination. Thanks.
But I think this one too.
Getting your volume. Just a kind reminder, hopefully everyone knows how to use the pipette, but if you don't know, this is very important. For a P1000-090 on the screen, essentially, or on the mirror, depicts 900 microliters. 010 depicts 100 microliters. When you're taking in a volume, go up to the first stop. When you're taking it out and placing it all into another tube, make sure you go to the second stop, because this is a problem that some people have experienced and they lose bacterium as a result. So please make sure when you're going in to take in volume, you go up to the first stop, you'll be able to feel a difference, and then go up into your other tube. You flamed it, essentially, aseptic technique. And when you're going down, go all the way down to the second stop. Make sure that you get all of the volume inside your volume. And these questions, I can scroll down and help demonstrate individual. You finally mix those. Get like a wrap. They may not have it. Thank you.
Yeah.
Done. Mixing, you can place them into this rack, or you can give them to me and I'll take them.
You have to leave the incubator first and then put it up.
We do.
Yeah, we gotta put it in. I was yeah, where did you microchip it? You're so fantastic. Oh, that's very cute. It's here. I'll find it.
Remember, you've labeled your two. And don't worry, I can slow down, I can go back to the full game. You can have these questions.
No, it goes in here. It was kind of fixed on here. People know what to do. Once I get my space, like I use sterile ones in here, and I dump them in here. Out. But I do guys put anything inside. They're already yeah. It's probably big ones. You got it, like dogs or anything. And it's subscribed. Yeah. No, you just scrape it on. I'm gonna stack her plate. Plate two.
I started using
We're taking all three plates together. So essentially, once your group is done with giving me your tubes for conjugation, we'll be placing them shortly into the incubator inside the second one, underneath the first one. And then you can go straight away to your exercise number 1 for treatment. If you have waste from the swabs, you place them into either here, essentially the microchip canisters or bottles, people say jugs or boxes, jars, essentially. You can place them inside, and that should be okay. We'll be talking about shortly exercise when you've already done, but for timing, you have about 15 minutes individually to do this class. When you're done, just have them all labeled within your group taped together, but have them individually recognizable because you have your own initials, MS, AS, MJ, for example. Just label them with your initials or name, including the other three traits I mentioned earlier: exercise, practical number 8, and the 2 of February 6th.
Anyone?
Yeah, added it next either or. Check it out. Then down all of it. If you find yours without color, let me know.
This conversation. How do I write it?
You pretty fresh. That's actually why they call that. Okay. Incubate all three. Give me just a moment, everyone. I'll answer your question. How many of you need extra plates for street plays? Exercise one. Please keep your hands raised. I just want to make sure I get enough for you all. One, two, three, four, five. Okay, let's see if I can get 6 for you guys. If you don't have plates for exercise one, let me know so that way we get more for you guys. You're not you have the luteus on your benches as well as groups. You can make use of them together.
But I would. But like, after. But at nighttime, right? Eat again. Water, and I.
Does anyone have extra inoculating loops on in their white trays, long trays?
Yeah.
Yeah.
Okay.
Yeah. Raise it up again. That's sterile water. Take your water. If you need sterile water, each bench should have a glass canister looking like that. That's sterile water with the hash. You'll label them individually. So you can actually have them all. And then just so you guys one and then.
And things are done, right?
Yeah.
No. We'll put it in that page. Whatever you want. Okay.
I have one here. I'll be only yeah. Yeah.
When you're done, I'll put them into. Yes. Try to complete in the next 6 minutes. I'll give you 7 minutes. Exercise 1. When you're done, let me know and we can move further ahead.
We have the others.
When your group is done, exercise 1, tape them up with your bench number and the exercise, and then we'll place them into the incubator all together. These are extra plates for those that needed them for exercise 1. You should have enough time. I'm going to restart the timer. 1 moment. Okay. So now you should be able to pass.
Yeah, that's right. Well, like, yeah. Know what I mean? Everything about the stand for that. Yeah, it's just-
If anyone has anything from the world, please bring it back because there's some other. So.
Yeah. Starts to get ridiculous. Right, this thing you actually. Like in a corner. I'm free. We do fasting for days. Yeah, differently. We only go for. It's hard for Greeks. It's like you're taking away our soul. It's not good. Oh, the doctor.
So when you prove this done, make sure that your button is off when you're not using it. But when you are and you've done exercise 1, let me know. I can place all of your group's plates inside the incubator. Well done. Just want to give you an update. You guys are doing pretty good, very well. So just keep it up, and you may finish early. So keep it up, everyone. Everyone, just want to remind you, please, when you're dealing with plates, just like tubes, keep them close by to your Bunsen burner. We don't want to have airborne cross-contamination of your bacteria. So just please bear that in mind, because otherwise you will see different colonies growing up.
Yeah.
Exercise one, just want to make sure it's a bit clear. For inoculating them, add a good amount, maybe a third if needed. This is a good range essentially of bacteria that you want to add, because if you go 2/3 into the agar, not sticking through but on the slant, you should get enough streak. So just bear that in mind, aseptic technique as well. And when your group is done, you can have them inverted, and I can take them accordingly. Actually, what it was, you know, so you
I know, I saw it. I was one too. I was one that- I absolutely stopped it.
Yes. The rest is fine. It's just right here.
Wait a second. Before you go. Did you already?
Yeah. It's 0.2.
Did you like,
Yeah. It's this way. Yeah.
Because if it
This
cuts,
is this way. Like this.
so you actually want them to connect. Look how pathetic it is. If there is an extra one, you could do it again. Is it clear? Go ahead with it then. One moment, I think she has a question.
Okay.
No, I think the number 3. Second line. Second, third line. Honestly, that's if I get the directions right. How do you do it? Where did you use the week before? Were you?
See.
Thank God. Everyone wanted to stay for time. But it was with Marcus. Just wondering, is it okay if I went into each section? But yeah, I kind of went into the A one.
Can I see those?
I don't think it's you.
Were you able to see it, or was the community making it?
The other look.
You had that.
Yeah.
So it's very hard for me to see here.
Yeah.
I can come to your bench with the.
Hopefully, in the next minute or so, you should be done exercise 1. After a few final questions, I'll talk about exercise 3.
Exercise 2.
No, it just may be a bit different. Essentially,
It's just I don't enjoy classes that much. Yeah. I go to like 50-year-old dad. I go to
I'm sorry.
Oh, yeah, I can label them. Yeah. And then she'll be. So
Pre-warnings and then on the deplates. So what you can do now is start labeling them, and then we'll place them into the incubator. Our incubator for practical 8 is in the second row, the blue one underneath, and it's at the top left side. I was just there, but for illustration, it's pretty much down at that end. And so I'll show you when needed. But the point is, when you're done labeling all your plates for the dilution factor, pretty much all 4 grades I mentioned before, except including the dilution factor, 10 to the negative 3, and 6. You place them not there, the second incubator, right underneath. Underneath. Yeah, to the left, and then pretty much where it's to you just went now. On the top left row or section is where you put your NYD. Then second incubator, see the brown one? Underneath that is another incubator, gray and blue. In the top left side.
Sorry.
In the meantime, before we go to the processing slide, you can have a few more things. I'll talk about it shortly. Also, anyone who hasn't done exercise 1, please raise your hand. Which group is finalizing? All of the groups are done. There should be 5 8 plates inside altogether, rows of plates in the second incubator. So just bear that in mind. We are a bit short in time to go straight ahead to the next exercise. So in the meantime, place in your NYT plates, and you can label them with what I mentioned earlier. Your bench, the exercise, number 2, and the 8.
In the bioreal. Yeah. Because I compare medicine.
This is for exercise.
Yeah, we're doing vaccine.
Oh, we put,
People are all set here. Yeah, these.
If you need extra pipette tips, we have them down outside. Just make sure that you let me know, and then you can place it in the box down there. Just make sure. Remember, when you open your pipette tip, the box should be close by your Bunsen burner, similarly the plate. Just make sure that you don't have anything else that would make it difficult for you to drop or actually control your pipetting. Anyone have questions, concerns regarding exercise 2, the plaque assay? I can go over it with you, but I just want to also answer any outstanding questions. Perfect. So let me just go over with you. I know we didn't touch upon it earlier. I said I was going to after each exercise. So this is the time to talk about the plaque assay. Everyone, I just want to make sure you're paying attention. That's for the conjugation. We have about a half an hour time period. So does everyone know where the items are located for the plaque assay?
What is the bacterial matter?
So
No, it's not really plus, it's not inside.
So these are the things that you need, everyone. You have your bacteriophage. Okay, even before we move forward, there's a few instruments that you have available with you that you want to bear in mind. You have your bacteriophage. It's labeled with lambda, right, in the ice bucket, if I, or sorry, in the smaller tray holders, like the orange one that Group 5 has. Each bench should have all of the necessary items. That includes your bacteriophage. It includes E. coli. Remember to resuspend when you're inserting in and out your volumes. And you should have extra tubes as well for you to complete today's project for serial diagnostics.
There's nothing inside to It's nothing. It's bacteria thing back. We didn't get any.
me run it down first. Okay, so you have two main tubes. One that's labeled lambda. That's the Greek symbol lambda for your bacterial phage. Your bacterial phage is already diluted and it's known from the lab manual as test tube number 2. So what you need to do is with the other epitubes found in those other aluminum foil trays right here. These are your epitubes to label test tubes 3 to 6. I'm sorry, 3 to 5. 3 to 5. Because you have other, you're going to go to test tube number 6 for the other dimensions. This is why I'm leaving this in a bit. If you haven't added anything, if you're sure. So just pay attention, everyone, because you have the test tubes already shown here. So I just want to make sure everyone's paying attention. You have the tubes that you need. You have extra tubes as well to label for dilutions of 10 to the minus 4 to 10 to the minus 6. That's test tubes 3 to 5 respectively. And so essentially you're going to do the dilutions like we did last week. Everyone, can you please pay attention? Everyone, I need your attention. So it's very important that as we move forward, that to reduce the risk of making mistakes, that we follow instructions accordingly. You have these test tubes that you're going to do dilutions of your bacteriophage. It's labeled as test tube number 2, meaning you're not starting with these two. You're going down from test tube number 2 all the way to test tube number 5. You have them labeled with new epitubes on your bench tops. And then essentially, you'll be making another set of 4 tubes that will have your bacteriophage included with 300 microliters of E. coli. You have a negative control, which in your case, technically speaking, is more like your positive control because you won't have any bacteriophage included. It'll just be 100 microliters of water. Sterile water shown in those glass canisters I mentioned earlier. The hashes represent sterile materials. So essentially, you have 5 tubes that you're going to make as step number 2, all the way to step number 3. And then, when you're done this step, you have here at 55 degrees, soft agar tube. That hopefully will answer the question made by group number 6, because these are the tubes that you're using. And when they're already prepped, all the way to step number 3, that's only when after 2 by 2, just for one sample at a time, you're going to be adding in the E. coli and phage mixture, all of it together, 400 microliters, right? You can literally use your pipette 040 and then add it in to this tube, the soft agar red one here. And then you just roll. Make sure your Bunsen burner is on, place it into your plate. That way, you just maintain not only sterility, but you maintain your bacterium with the bacteriophage in your NYZ plate. It's going to solidify after some time. After that, then you invert it, and you can repeat the process for your other plates by your other group members. Don't forget, you label your plates, plate A to E. They have different conditions. A is your negative control. B to E is the ones that you have their diluted bacteriophage with the same amount, same volume of coli.
No, I have a second question. My second question is.
Everyone pay attention. I'm going to answer questions so that way I'll help everyone has a very similar question.
Okay, so in the lab manual, you said we start out at 10 to the negative 3. Is that just not the case?
That is your lambda tube. That is your test tube number 2. That is 10 to the minus 3 already. You're assuming that. You don't have to make it. It's already prepped for you, and you're making the next dilutions for 10 to the minus 4, 10 to the minus 5, and the minus 6. I should know. You said it's time. When I said last name,
Yes.
Wait, just here for a moment. Have someone also keep track of time, because they'll be timing it. Sure. Sure. That represents the bacteria. Now, hopefully everyone started in the for the end of the assay. When you're done and you want to actually incubate your test tubes, all of them, then what you do is essentially place them inside.
Yeah, so these are our problems.
So please bear in mind you're adding in the right volume. Some of you may have been going to the second stop when you're aspirating in. That's why you're taking up more than 300 microliters of E. coli. We technically don't have enough to share with everyone because we still have so if you're having let me know.
You can look. Ironic. Yeah, my name is like sad, but like nothing else. I'm doing everything. Good fortune.
My name is I don't look like it's like sort of stuck. I just
So we got the Ebola.
No.
This should be up to 7. You could take this in the meantime. You already has the orange one. Either way. When you're done, you can
Put it back, right?
So what are you guys doing so far? Are you guys done?
They're doing exercise 3, I'm doing exercise 2. Exercise 2. I mean,
Like, I don't want to also work in the most efficient way. How things are going on. And you guys are like one of the telling, keep it up. So keep
We have questions.
Is anyone or are we supposed to do that ourselves?
You're supposed to do it for the information.
For the 2 hours.
I'm
Thank you.
11 minutes. Technically, we can go up to an hour, like an hour to 2 hours. So what that means is essentially when you're talking about it, I would say first focus on the plant assay as your part, and your group can focus on the transformation assay. Then you can go to the plant assay. So just let me know so I can keep track of wherever.
Okay.
I think so. You can wait a bit of time. You have a bit more time. The more longer you wait, better, actually. But I would say complete what you're doing and see if you can finish in the next 20-ish minutes.
All right. Greatly appreciate it.
So how's everyone doing so far? Please do not have phones open if you're not using them for any scientific reason or generally speaking, an appropriate reason. You can let me know, but you just need to make sure that you're paying attention. I also want to say, how many of you are at stage or step 3 of the plaque assay? So how many of you are about to place their tubes into the incubator? Raise your hand. Because there are 2 groups that haven't. Your group has? Are they in the incubator? No? Okay. Your group, sorry. Okay. Group 3 or group 2? Have you guys? Okay. Okay. So I'm going to give you about 15 more minutes to complete the plaque assay. If your group is in the meantime almost done, and 2 members of your group can focus on the plaque assay, I recommend the other members to focus on transformation. For transformation, I'm going to change the slide. The idea is the following. You're going to be using the plates that have been spread plated or streak plated in the middle bench here. So just please pay attention, everyone. For transformation exercise 3, you'll be using these plates. You just need to take 2 colonies, and if you find it difficult to see, try to use another plate. But remember to please bring them back. So they should be 6 in total for 6 groups. You have LB broth also here if needed, but you have as well the sterile pipettes that you could use, which are typically used for microbiology. Now, please bear in mind, for timing, in general, I want to make sure everyone is up to pace. So, in the next 15 minutes, please be complete your lab assay. And this will be important for your formal lab report, due on the 1st. So, please bear that in mind. And we'll talk about a few other points for the upcoming exercises. But I want to make sure that you guys are done very soon. 15 minutes at max. The timer is going off for the conjugation experiment. So first focus on the plaque assay. Split up to have members of your group focus on transformation. And then at the very end, before 11:25, i.e. half an hour from now, you could go back to conjugation and complete your 3rd plate. And we'll place all 3 plates together into the incubator. 8%. Wow, where do we incubate? Your ice bucket has what you need: calcium chloride, 1, 2, CaCl2, and the plus is your plasmid. So just bear that in mind. You have the plasmid that you need. It's in your ice bucket. Remember, for heat shock, you're going to be adding them into here for 42 degrees. Remember, based on the manual, that there's very clear instructions on what you need to do. But if you have questions, come by me and more than happy to help. Actually, you have a weird. No,
What is the tape?
if any group has extra tape, can you please? What did you want? I would recommend for after one. Generally speaking, you can keep it if you need to. You can keep it.
What is 42?
Okay, two good. I'll help you. Both will be together. So you can place it into the second. Take it out one at a time. Follow roll a bunch, and then be able to pour it again. With your ARC, otherwise you can the incubator.
Yeah.
Just a plus. It's
Yeah. Those have. Okay, go for it. A little bit, I don't know if it's measured.
This yeah, we don't do it.
So it's recognized. So that's all there is.
It's from over there.
Carlos, 400 microliters, right?
Yes.
Give me. Anyone needs extra E. coli or extra lactate for the podcast thing? Please let me know now. We're gonna ask lactate. Otherwise, please go ahead with that information. Once again, we have things here for the bacteria E. coli HB101. You have it here for transformation. Continue to move on. And plasmid and
It's one of the.
So yeah, we get Alameen.
Okay.
Yeah. Oh, I'm not. You look amazing. You look amazing.
Who's doing the? Guys, can you?
Where's the start, please?
Thanks. Sorry, we did it wrong. It's not supposed to be. We need a different one for each one, don't we?
The negative one for the negative, the negative one for negative, positive. And then we use the last letters.
Okay. They're there. Oh, yeah.
So we need to off paper.
It was around.
So, just a quick question. How's everything going for everyone? Okay? Okay? One group is asking if another group has extra lambda. They need about 70 more microliters. If you have more, please let me know. We can group them all together. Thank you.
Yeah, can we bring this plate back and then before the you are.
Thank you.
We're supposed to do, what we're supposed to do is, take one, mix it inside this, mix it, pour it inside of it. There's 50 seconds.
Jacob, no, don't ask for. Yeah. Thank you.
So once your group is done pre-warming your NYC plates for the plaque assay. So let's just go round by round and confirm these details for you all. The plaque assay, just bear in mind that you've already pre-warmed your NYZ plates. Many of you are likely in the mode of already doing transformation, but just to reiterate this for everyone. The NYZ plates are pre-warmed. You're then after for one plate at a time. It could be each group member, or ideally one group at a time, essentially takes out a soft agar plate, dumps in all 400 microliters into their soft agar, rolls it up, place it into the NYZ plate that's already labeled, and then all of them, once all of them are done, you could place them into the incubator. Label them with exercise 2, the practical number, your group number, and the date, as well as the dilutions that are mentioned here. Yes. You are actually
Okay, this are done. Over there.
Oh.
It's all turn. So we for real. But this. Here. Guys, let's invite it.
So, congrats everyone. I know it's a bit hefty. I would say honestly, a lot of things came up that were a bit unexpected, but nonetheless, very happy to see all of you excel. A few important things. One is obviously when we clean up, please make sure to bring back all of the equipment that you had earlier. The soft agar tubes can be placed in the middle bench. The HB101 street plates also have to be returned. And into the incubator, remember that you've labeled all of your exercises per bench and the street plates by your name. Now, when it comes to conjugation, we should be ready now to complete the last exercise right now. So what I'm going to do is take out your plates shortly. But before I move further, how many of you are complete the plaque assay? Raise your hand. Great job. You guys are done. You place them into the incubator. They're done. You guys are done. Your group is done. Almost done the plaque assay. Almost done. Okay. And does your group have anyone focusing on transformation now? How many of you are now working on transformation? Any group done transformation? Okay. So here's what we're going to do in 15 minutes. Have transformation complete because there is a 10-minute wait period. So what you could do is, in that time, you can actually start focusing on two things. Your spread plates, counting the colonies, but more importantly, the conjugation. I'm going to take out the plates, the tubes from the incubator. But remember now, it's very simple. You've made this tube. Just don't forget, resuspend your bacteria. Make sure that you resuspend them well. And then you just take one drop, repeat the process you did for the past two plates, now into the labeled plate 3 with both antibiotics. And then you should be able to smear with aseptic technique with the sterile swabs easily. With that, if there's any questions, let me know. I'll take them out shortly. And let me know if you have if there's concerns as well. Thank you. Remember, 42 degrees for heat shock. We have this ice bucket, also has the plasmid and calcium chloride. Yes, maybe just one from the right.
Yes.
What's degree? 42? For room temperature, you could actually just place it on your bench. But if it says 37 degrees, then it's a bit right. Okay. Actually, is it okay with
I think I'm also gonna need the album.
Yeah.
The last. So I'm gonna wear that. Oh, they're for my we're good.
Yeah.
Oh, yeah.
Now I'm just confused. Oh, can I have a look at it? Here are the tubes, the black tubes for conjugation. You can take them when you just don't forget. We'll talk about
Yes, yes.
That one. Yeah. Okay, and then just one, what do I see? Sorry, bless. Yeah.
So actually,
We need a music.
so
Yeah. No, no, we need
So, just want to get an update. Hopefully in a few minutes, you should be done both the plaque assay and or the transformation exercise, if you haven't done it already. How many of you are done transformation? Okay. Are you guys almost done? Okay. And okay. And the plaque assay, is it complete? Okay. You guys?
We're having the talk there.
Okay. How many more do you have left?
We just started.
Just started?
Yeah.
Oh, I see.
Should be on site.
So in about 5 minutes, I'm going to need you to start at least focusing on conjugation, meaning that those other two exercises for transformation and the plaque assay should be done. If a set of members of your group, please pay attention, everyone. If your group members are done one of those two exercises, please make sure also your Bunsen burner is not on too high. Chana, is your Bunsen burner on high? Yeah, but just make sure it's not too high, because I'm smelling smoke from someone. It may not be yours, but if anyone's Bunsen burner is on too high, please lower it. Hajra, can you lower your Bunsen burner just a bit? Okay. Now, please make sure in the next 4 minutes, you should be done transformation. If your group just started using the soft agar, make sure everyone's helping to pitch in. Make sure everyone's aware of what they need to do. If they can't do transformation and they're finishing up the plaque assay, they can start placing their plates once complete into the incubator. Top left of the second incubator. Sorry, I'm just smelling the air. Apart from that, let's make sure that you can complete the rest of today's exercise with ease. So remember, I'll put on, actually, remember to use the counters, your timers, to make it more experimental for yourselves, to focus on timing.
Number 5.
Okay, any questions? Sorry, can you repeat it? I don't think she's using sterile line because, well, I mean, these are the bacteria that you're using. Okay, transformation. So for transformation, we're using the HB101 colony label. You see those pleats?
Wait, you're gonna do it twice. We're gonna do it again for the other thing as well. Yeah. This is 400.
So if your group is almost done, let me know because I could give you actually your spreaded plates from last week. That's one of the final things for today. And you just need to record. Spread plate. Before I end off with the post lab, what are there other things? So in terms of what you have do you know what you have include right for your figures? Sorry, for your observations for your results? In terms of what you've done, public spread counts from last week, which I'll give shortly. But apart from that, also in terms of notes that you're going to be making from the exercises you've done today. Transformation, conjugation, the plaque assay. If you made any mistake, you will note down.
Same thing.
Please, I'll be placing them here in the middle bench. Only if your group is done the other exercises, then I would recommend coming here. If you're unoccupied and just waiting for timing to go, like for 5 to 10 minutes, you can also have this kind of stopping on.
For the bacterial propagation.
So you know, bottom left, top left.
Brown.
Okay. Just make sure you put the exercise.
Good. Thank you.
Yeah.
So,
Terry, what did you say? Okay, because that's what I thought. But you couldn't stand.
Wait, should we take our exercise too?
Yes, we can.
Just gonna answer this question about spread plates and counting your colonies for your post lab. So please pay attention if you're not doing any of the other exercises, transformation, conjugation, or plaque assay. Let's say you have, let's go back. I think it was, okay, here, for example. Now, let's just get the context of this understood. If you have a plate at 1 to 1,000 as your final dilution, because you took it from a tube of 1 to 100 with 0.1 mils, if your plate has a dilution factor of 1 to 1,000 or 1 to the negative 3, 10 to the negative 3, I mean, and you have colonies that appear as circles like this, I want you to count only plates that have colonies between 30 and 300. If you're having difficulty counting, just let me know. I could come and tell you which plates to count, essentially. Once you get that value, let's say it's 45, and your dilution factor is 1,000, hypothetically. Other plates may not work as well as at 1,000, but if it's 10 to the minus 5, it may work. If, let's say, your value is 45 colonies, then you multiply that by the dilution factor, not 10 to the minus, but 10 to the 3, which means that you have 45,000 cells per mil, or CFUs, which are colony forming units. That is your expected original density, cell density. If you have two plates that are essentially with dilution factors within 30 to 300 colonies, then you average the final original cell densities, and that's what you expect to be how much you had, or how much you would have expected in your tubes themselves, and in your original cell culture. This is beneficial from pathobiology because let's say you wanted to know how many cells are infecting a certain person, and this is how you're able to hopefully deduce certain logics from their plates if done essentially in this way. Yes. For conjugation, you counting them?
Yeah, we are.
How many?
Oh, yeah.
I wouldn't recommend counting them as you all know, because unless you're stuck with that,
Yeah,
you can. But I highly recommend because
but we also. Yeah. Because like.
okay, so let's take a look. Like, can you count this one?
We have.
Manually? How many people? Okay, if it's within that range, 30 to 300,
No worries. She's so funny.
Are you guys gonna be okay?
Yeah, I'm gonna ask them how to do it.
I'm gonna head out. Thank you.
Please, good luck. We're just asking how to count.
10 minutes or something.
I don't think so. But ours is stuff. They didn't have. They actually I think there's something. I'll count one, you count one. Write down. This is our okay. Thank you so much.
So before you leave, there's a few important reminders. And I'm actually very amazed that you guys are finishing like ahead of time. I really appreciate your support. And I apologize for the hecticness of today's practical, but I hope it is of benefit for us all. Before you end off, make sure you've cleaned up your bench. Make sure you've returned back the plates, HB101 plates, back to where they were. The soft agar tubes you can place. Actually, we're going to put them at the very back of the row of the room, so just bear that in mind. And in terms of your formal lab report, your formal lab report is going to focus on the plaque assay. Please pay attention, everyone. You have your post lab. Remember your results, calculations of OCDs, original cell densities for your 2 or 3 plates, making notes of your observations of the transformation, the conjugation as well, if you made any mistakes, and the plaque assay. But then on top of that, besides the questions here, you have to start preparing for your formal lab report. What I always recommend everyone is to essentially, please pay attention, group 4 and 5, please pay attention. I'm talking about the formal lab report. It's very important that you actually start preparing now. You have reading week. I may not be here next week, but you need to prepare now, because that way you're able to not only ask me questions early in advance, but that way you don't get stressed out with other term tests and assignments. So that's my honest advice for you as an undergrad of the past. And I don't want any of you being stressed out or having to do any unauthentic breaches of authenticity in our labs. So just bear that in mind. Before you leave, please make sure that you return all materials back to where they were before. Make sure you've labeled your tubes, yes, but especially your plates into the incubator for the exercises that you have completed. Apart from that, have an amazing day. If you have questions, let me know. I'm just going to skim by, see if there's any other. Oh, yes, very important. Try to complete this if you can. I know you don't see the results yet, but finish the first column at least. And oh yes, and lastly, slide 10 is where you actually really need to pay attention to the critical thinking questions. With that, Barakallahu feekum. Really appreciate the time. Have an amazing day. If I don't see you next week, have an amazing reading week. Take care, everyone. These are your spread plates of spread plates from last week. Spread plates go into the autoclave. Autoclave is right beside the fuel. Right on the right. Yes, God. Actually, I will make sure that also. So I now it.
Thanks, guys.
Thank you, Sarah. I'm over here. I'll do that. Y'all.
So, well, basically, I'm just start the 10 minutes. Stop. It's been like, what do you think? Doesn't matter.
You're ready to go. If you're done, and if you clean up your bench, you're ready to go. Thank you so much. Is the group missing a nucleus on a bench?
Okay.
Don't worry. Just do your best. The rest of the best.
The negative.
Actually, per time. Two separate weeks.
Oh, yeah, I was like thinking.
You just like the last one.
So. Have an amazing day. Just try to find it and keep the function very well. Because if you have, I really think that you have to on the volume that that's okay.
Okay, we need the plus, right? So,
yes. Just make sure you flamed your L. Like actually continue as you are. Continue. It should be okay. Everything okay? Okay. Let me help you guys.
My theory. They teach all these, but they don't really they didn't go as deep as you had and eventually alcohol. That makes it like a little bit I'm not quite used to it. And started out here in Borgo, my box. Now,
There's a lot of questions. Like, I'm imagine it's stressful. So I'm like, I'm putting yeah, spread. I don't know. Yeah, it's very painful. Is it possible to start this week or no?
Yeah, you should.
Because you need to know it's about this week's side, right?
Yes, for the plaque assay, but essentially you have reduction.
No, it's just like background stuff.
Yeah, but it helps because you don't have to worry about it down the road. But in part, pretty much in you'll be able to essentially count and get those observations into the formal lab report. And then in reading too much focus on the discussion. You just have to input them onto either Excel or Word, and then you should be essentially complete message of a lab. Intro could be this week, results next week, and discussion reading.
Okay, I'll try that. It's on March 1st.
No, but I just the reason I say that is because often you think like, oh, it's so much fun, but if you do it well, even 3 hours per section, you could get a really good grade. You just have to put in like everyone I want them to put in the right effort.
Well, 100% I will. I have the third song.
Oh,
That's expensive.
okay. See if you can then after or before.
Next week, right after that, lab, and it's for like an hour, so what's one, I'm also gonna have to that night.
Yeah, I think you can technical.
Yes, I think,
You can hold up one like I don't say it's next week or
yeah, next week. Probably fine.
please I would say you not want to run, it's not enough. She may have to do, but otherwise it should be. Also like it's not multiple bacterial. So just in case.
Where should I put these?
They would want to.
South, for brown, like brown, right before the summer. Yeah.
Take care, everyone.